Hello, everyone. So our project is decoding inner speech with brain-computer interfaces, and it is a study in EEG analysis and machine learning. Our PI is Sahar Jahanikia, and the presenters today will be Denise, Ritwik, Shravani, and Jungkyun. So an introduction into inner speech and the dataset we have used. As I sit here, my inner voice is constantly chattering, giving me a running commentary of the world around me and my actions. It is a complex and ever-evolving stream of consciousness that ranges from practical observations to abstract musings, and it can be both a source of comfort and a source of frustration. This is my inner speech. Inner speech, also known as verbal thinking, refers to internal verbal dialogue that we engage in with ourselves. It is a form of private communication, and it is thought to be an important aspect of human cognition because it allows individuals to plan, reason, and reflect on their thoughts and actions. Inner speech can be spontaneous or intentionally controlled. Research has suggested that inner speech involves similar neural mechanisms as those used in external speech, such as activation of similar areas, specifically the Broca's and Wernicke's areas of the brain. The Broca's area, as depicted in the picture on the bottom right of the screen, is used for speech, whether that be external or internal, while the Wernicke's area is used for comprehension of what a person reads, sees, or thinks. Other studies suggest that inner speech has distinct characteristics, such as faster pace, greater variability, and less systematic relations with speech-related neural regions when compared to external speech. The process of inner speech is a dynamic one, with different regions of the brain interacting with one another, both flexibly and adaptively. For example, when we are reflecting on our thoughts and emotions, the self-referential processing network becomes more active, whereas when we are actively engaging in problem solving, the frontal regions of the brain become more active. Inner speech is thought to play a crucial role in a wide range of cognitive processes, as mentioned in the previous example, including memory, decision-making, and problem-solving. It has also been found to play a role in developing and maintaining mental disorders, such as depression and anxiety. Inner speech is connected to other neurological activity, such as spatial location or movement, including visualized condition and pronounced speech. Now, brain-computer interfaces, BCIs, are systems that allow individuals to communicate or control external devices using their brain activity. Inner speech has been proposed as a potential source of signals for BCIs because it is a form of verbal thought that the individual can directly access at all times. This topic is the basis of our research, but it is important to first take a look into the dataset we have used to conduct this study. The dataset used in this research contains data from a study that aimed to investigate how neural signals correlate with inner speech. The dataset comprises electroencephalography, EEG recordings, obtained from 10 healthy right-handed participants with an acquisition system of 136 channels. The sample included 4 females and 6 males with a mean age of 24 with a standard deviation of 10 years. All participants were native Spanish speakers and did not report any conditions or disorders that could interfere with the research. The participants are identified by aliases, sub 01 through sub 10, as can be seen in the pictures on the left, during the study. The study was conducted with 3 consecutive sessions, each comprising of 5 stimulation runs corresponding to different conditions: pronounced speech, inner speech, and visualized condition. The total number of trials varied depending on the willingness and tire of the participant, but on average, each individual performed 200 trials per session. There was also a baseline interval at the beginning of each session where the subject was directed to note, move, and relax. For the pronounced speech, the subject had to repeatedly say the word aloud to more clearly define the motor regions which were activated. In the visualized condition, the subjects had to focus on moving a circle in the center by the directions indicated. The raw EEG data was stored in BDF, also known as a glimpse bitmap distribution formation. There were also 8 external electrodes used to measure artifacts that were disrupting the electrical signals, and those were saved in FIF files.
Now, I will be going over background information. This will help you understand material that will be covered in their sections. So first, what is electroencephalography? Well, it's a relatively new neuroimaging technique with the purpose of recording the electrical activity of the brain with high temporal resolution. It does so via electrical devices called electrodes, and usually these devices are placed on the scalp of someone's head, and thus they're able to record the electrical activity that happens because of neuronal interactions. It's commonly used to diagnose disorders, and this is because of the specific patterns that can be seen in the EEG data that can indicate such disorders, such as strokes. And as mentioned before, it can take advantage of the nature of neurons, specifically pyramidal neurons. On the right, you can see a sample of how EEG readings look. So you can see the oscillations of electrical potential. And on the left, you can see how the electrodes look, and you can see them being placed on the scalp and the pink region signifying. So now, how does EEG specifically work? Well, there's four main steps. First, neurons communicate via electrical chemical signals called neurotransmitters. And when these neurotransmitters bind to a postsynaptic neuron, postsynaptic means that the neuron is receiving the signal, and presynaptic means it is sending the signal. Now, when the postsynaptic neuron receives the signal, it can cause excitatory and inhibitory postsynaptic potentials, or EPSP and IPSP, respectively. The response causes the electrocellular space to polarize due to the resultant dipole, and because of an influx of cations or anions, depending on the specific scenario. These ion currents are formed due to the resultant dipole, and when this activity is synchronous with other pyramidal neurons, electrodes on the scalp surface are able to read the resulting electric potential. And this is because it becomes strong enough for the electrode to actually read. On the right, you can see a diagram of this. The black lines signify a thing called volume conduction. And the basic principle of it is when the electrical potential goes through different biological materials, such as the skull of someone, and it reaches the surface of the scalp. Therefore, the electrode on the surface of the scalp is able to read the electric potential and its alterations. In addition, the yellow signifies an electrode on the surface of the scalp, and the red signifies a dipole that would be made. On the right side, you can see an example of an EPSP and IPSP going from left to right. An EPSP causes an influx of positive ions. Some of these tend to be stuff like sodium, and this causes a negative electrocellular polarization. This can cause a positive polarization more near the pyramidal neuron cell body, and thus create this dipole. The same effect is with IPSP, but it's just reversed, where near the postsynaptic neuron, where it actually had the neurotransmitter bind to it, it's more positively charged on the outside and negatively charged further up. Now for EEG channels. As mentioned before, EEG uses things such as electrodes in order to record electrical signals at various parts of the brain. And a channel is an electrode at a specific position on the brain, and it's usually denoted by two things. First is by the letter, and this denotes the location on the scalp. So for example, those towards the center tend to be called like midline or vertex, while those in other parts of the brain, such as occipital and temporal, are placed around the other parts. Next, we have the number, which is the distance from the midline. You can think of the midline as a vertical line in the center of that scalp diagram. And those with higher numbers are farther away from the scalp, farther away from the midline, and those with less numbers or those with Z are closer and on the midline respectively. You can see that on the diagram to the right again. Those at the midline have Z as the second denotation, and towards the outer areas, so for example, for mid-temporal, you can see the numbers getting bigger and bigger. It is important to mention, though, that not all channels have to record brain data, although most of them typically do. Examples of these are things such as the electroocularogram or EOG. This is used to record eye movement since they can cause deviations in electric potential, which are easily seen and noticed on EEG data. Another one is no neural activity on earlobes. So this is when you place electrodes there to capture any activity which is non-neural. This is especially helpful because when you are filtering the data, as will be discussed later, you can just filter out based on the activity recorded by these electrodes. Now for specific artifacts. Now, although electrodes can record brain data, they also record things which are not from the brain. And there are two main types of artifacts. First is physiological, and this is caused by body movements. So as mentioned before, the eye, the reason why eyes cause deviations of electric potential is you can think of them as a dipole, with the cornea being positively charged and the back of the eye being negatively charged. Therefore, when you blink, your eye moves up, and thus that deviation potential is caught by the electrodes. You can see an example of this in the top right image, where the red boxes outline these deviations. Other examples are muscle artifacts and cardiac artifacts, among others. Non-physiological artifacts, however, are caused by mechanical electrical equipment. This is due to the alternating current, which is used to power electrical devices nowadays, and it can also be caused by other factors such as the environment. The significance of this is that it contaminates the data, and it needs to be removed by methods such as filtering and independent component analysis in order to make sure that our data can be used for BCI methods and later processing steps. So I've already discussed the image on the top right. Towards the bottom of that, you can see a picture of line noise, and this is the noise which is caused by the mechanical electrical equipment. Notice how this data has way higher frequency than regular EEG data, as you can compare with the image in the top right as well. Now for introduction to the brain-computer interface. Brain-computer interface is a software that interprets signals and uses translation algorithms to convert the signals into commands that an external device can use. The goal is to create a direct connection between the brain and an external device, allowing humans to control devices such as wheelchairs or prosthetics with these thoughts. For example, if someone has a stroke, this can cause things such as impaired movement of different joints, for example, of upper limbs. And since, obviously the movement of your upper limb is needed for day-to-day tasks, things such as BCI can help because it can find patterns based on the person's thoughts and thus can move based on what the person commands. Now, there are three main types of BCI. First, invasive, which involves the implantation of those EEG electrodes directly into the brain. This type is most accurate, however, has a higher risk of complications. Non-invasive, by contrast, is when the electrodes are placed on the scalp, and commonly this is the type of EEG and the electrodes are actually used, and this is what we actually use non-invasive BCI for our research. And partially invasive is a mix of both. Finally, for the BCI structure, now how to actually construct a BCI comes through 5 main steps. First is the signal acquisition, which is when you have an EEG recording in order to gain the signal and the electrical potential deviations from electrical brain activity. Second is signal preprocessing, and this is when the signal gets cleaned up to remove any artifacts, as mentioned before, such as eye artifacts, muscle artifacts, channel artifacts, etc. And this can be done through steps such as filtering and ICA. Third is feature extraction, and this is where EEG signals are given a distinguished core set of values. Now, because lots of EEG recordings are continuous, it is important to only take the parts of the data which are needed in order to construct the BCI. This is based on when events happen or when stimuli happen mainly, and that's where we can do things such as epoching in order to get these specific parts. More on that later. Fourth is signal classification. So the signal gets categorized based on the values given by the previous step. So for example, signals which are in response to the stimuli of a person thinking of the word right would be given one classification, and then signals giving of the person thinking of left, for example, would be given another classification. Finally, computer interaction is when the appropriate algorithm is applied based on classification, and this is things such as BCI prosthetics, which can be used to move limbs, which a person no longer has neurological control of.
All right, so now I'll be going over the methodology of our research. We distilled the different stages of our research into 4 steps. First, we researched EEG lab and different EEG preprocessing tools. Next, we preprocess and analyze the inner speech data. And finally, we plan to develop a machine learning model to decode EEG signals. Okay, so now I'll be describing each step more in depth. When researching EEG preprocessing methods, we looked into different standardized preprocessing pipelines, such as the Harvard Automated Preprocessing Pipeline, known as HAPPY, and PREP, which is a standardized preprocessing pipeline for large-scale EEG analysis. The objective of these preprocessing pipelines is to standardize the process of removing noise from EEG data since this commonly differs between researchers. These pipelines outline distinct steps that are necessary to complete before actual analysis, and an example of what these steps might look like is on the flowchart to the right. Some of the steps include things like filtering, channel rejection, and ICA. During this part of our research, we also looked into EEG Lab, which is a MATLAB toolbox, and EEG Lab contains pre-written functions to implement pre-processing steps, which makes it really useful for us. After we had a strong foundation in EEG pre-processing methods in EEG Lab, we began to pre-process and analyze the inner speech data. For consistency, we decided on a set of steps for everyone to follow. The first step was to delete any noisy channels, and next we filter the data by excluding any signal below 0.5 Hertz and any signal above 60 Hertz. Then we resampled the data from 1024 Hertz to 128 Hertz, which reduced the size of the data set, making it easier to manage and also faster to run algorithms on. And then next, we used automated artifact rejection, which removes intervals of the EEG recording that are likely to be noise. And after that, we used independent component analysis, known as ICA, to separate the EEG recording into different components. Since that's sort of abstract, on this slide are some examples that might be helpful for understanding ICA a bit better. In the image to the left, what ICA does is it takes an audio recording from multiple sources and separates it into its constituent voice recordings. And similarly, ICA can take an EEG recording from multiple channels and separate it into different components. So after running ICA, we extracted epochs from the data, which allows us to analyze brain activity associated with a particular event during the EEG recording. The results from these steps will all be discussed in the next section. Our last step is to develop a machine learning model that can classify different EEG signals by what the subject was thinking. And one thing to note is that this step is currently in progress, but for machine learning, for EEG data, it can be categorized into two approaches. The first is feature-based methods, which extracts features from the data, which are then used to train the model. And the other approach is end-to-end methods, which feeds continuous EEG recordings into the model. A comparison of these two methods is shown in the image here. And typically, end-to-end methods use deep learning or some other type of neural network, whereas feature-based methods rely on other types of machine learning. To build our classifier, we are planning to use BCI Lab, and like EEG Lab, BCI Lab is a MATLAB toolbox, and it offers many different types of algorithms like linear or logistic regression and support vector machines, and it also has many feature extraction algorithms for common spatial patterns and covariance features.
All right, I'll be going over the I'll be showing the visualization that EEG Lab has produced for us and kind of like show you what the progress we have made with pre-processing so far. So firstly, this is one of the data visualization tools we get from EEG Lab. It basically shows the channel, the location of the channels. So as Rick explained, there's a lot of channels for EEG, and for our data set, we have 128 channels, and then 8 additional external channels. As you can see, this image here is not actually unique to our study, as studies that use the same EEG machine would have the same channel locations. But this information is used for drawing spatial significance from the EEG recordings that we have. This is the raw EEG data that we have. The image here is pretty straightforward. It shows the data of our EEG recording. You can see that there's 128 lines. There's a lot of lines, and there's also 8 additional lines at the very bottom. This visualization was also what we had available for us to perform manual artifact rejection and channel rejection, and I'll go over that later on. In total, we had 30 of these files. 3 sessions for 10 of the subjects. Each session is around 1600 seconds, which is around 27 minutes. And there are 1,000, as I think Denise explained, we had actually, explained we have events which were just marked time places where we had specific events going on. So that'll be stuff like start of protocol or start of a specific treatment. And EEG Lab, through giving these visualization, allows us to easily and manually pre-process our data. So what you see here is a channel spectra map. This graph uses several mathematical functions like the Fourier transform to decompose the EEG data we have in all 128 channels to the different frequencies that overlap. The EEG machine is a very sensitive one, and it collects all the electrical signals from the brain and the environment around it, as Rick explained. And therefore, that means that there will be a mix of multiple signals, either from the brain or from the environment. So the brain gives off like different types of signals, so different types of waves with different frequencies, such as alpha, beta, delta, theta. And these all have different frequencies, but on the EEG recording, they're all added together. Now, for instance, we're going to have signals that are 20 hertz, 15 hertz, 8 hertz, and maybe a 50 hertz signal from the electrical line noise. Instead of having 50, like 4 of these sinusoid waves separately, we're going to have all of them added together. And it's going to be hard to tell them apart visually by the human eye. So what a channel spectrum map does is it uses mathematical functions to kind of show us which frequencies are present. Now, on the image on the right, this was a spectrum map for subject 1, session 1. This one shows a peak at 50 hertz, which is most likely line noise. This allows us to kind of get an overview of what brain activity are available, and also perform pre-processing and check which artifacts are present. Okay, we were able to run pre-processing all the way through on 2 of our subjects, and there are 3 sessions through EEG Lab. As previously explained, this process included filtering, artifact rejection, independent component analysis, component classification and rejection, and data epoching. To demonstrate why the EEG data visualization is important, the image on the left shows channel D8 through 11 are abnormal and full of noise. That will be kind of hard to see, but it is on like the third quarter, third quarter down. There's like a bit of a darker region of the EEG lines. That's channels D7 through D8 through 11. For easy viewing, the image on the right is actually a close-up view of those, and you can see that there's a lot of noise for those channels. Now, for D8 through D10. Now, what we had to do was reject this. We actually had to visually go through the channels to see that these channels were actually malfunctional, and we can't use the data for these. Therefore, we just basically removed them from our data set and had to run pre-processing without them. That wasn't a big problem since there's a lot of channels dispersed throughout the scalp, and we could have used data from other channels. Okay. Okay, so independent component analysis, as Denise mentioned, is a technique in neuroimaging signal processing where we extract spatial and temporal components for further analysis. This is run through the preset algorithms on EEG Lab. And for us, as of now, we are using the run ICA algorithm on EEG Lab. The image below shows the different components produced by our run of ICA. We had to manually go through the labeling and rejection of these components to move on to our final steps of epoching and machine learning training. So this is, I believe, the ICA components for subject 1, session 1, and in total it produced around 130 components. After we had to manually go through and reject some of these components, we had around 88. Now, there are paths for doing it automatically. For instance, the IC label algorithm also labels them, labels the components, but it is not perfect, and therefore we still had to go through each of these components to reject them. The image on the right actually shows one of the components that we would have rejected. IC label has resources on which components to reject, and that was one that we primarily referred to for rejecting these components. So component 74, for example, has a big, red region on the side, and this kind of this is most likely channel noise. So this would be one of the components that we would have rejected. Lastly, as Denise mentioned, data epoching splits the data into different segments that surround a certain event. So we had around 1,000 events, and basically data epoching cuts out every time segments that are not around an event, and we have a few seconds before and after the event, so that we know we only care about the brain activity before and afterwards, to see what the event has done to the brain. And this data would be used later on for machine learning. Now, talking about what we have planned for the future of our study. Sorry. Yep. We are planning on finishing the entire steps of pre-processing and machine learning training with the first two subjects, just so that we have a full sense of what we have to do, so that most of the troubleshooting happens now instead of later on. And we're working on, since we already pre-processed the first two subjects, we are working on putting that data on BCI lab, so that we can finally develop a BCI algorithm. After that, we're going to apply what we learned and the steps that we took on pre-processing all 10 subjects and putting them in BCI lab to develop a machine learning model. Lastly, we will hopefully have a brain-computer interface that will be able to tell what a subject is thinking in their inner speech. For her unwavering support and guidance, this project would not have been possible without her expertise and supervision. Additionally, we would like to express our sincere gratitude to ASDRP for giving us the opportunity and resources to carry out the research. Lastly, I think it is important to acknowledge that the four of us are not the only ones who did this research. There are other members of our team, and they also contributed greatly for the research that we presented to you guys today. So we would like to thank the other members of our group.
Mm-hmm.
Thank you so much. Wonderful presentation. Wonderful. So well done, well done, well done. Amazing work, amazing progress. So proud of each and every one of you who are presenting, as well as the one in the background that are working in this project. So I think they're open to questions. I don't have anything other than just saying that how grateful I am.
Yeah, I have mainly two questions. First of all, I would like to congratulate you and all you guys working on it. It's a wonderful. The presentation was so clear, and then slides were clear. Everything wonderful. My question is that you might get the final concrete result. So who will be benefited from your result at the end? Who's beneficiaries? One question. Another one is, are you guys going to continue this project for future as well? I mean, in the BS or maybe MS or PhD, are you thinking to continue this project for your future university?
I can answer.
Yeah,
sure. Yeah, I can answer these questions if no one else. Okay. Yeah, so BCI, as the name implies, is a interface between the computer and the brain. And what this primarily refers to is for people who do not have a proper connection with, so there's two ways that we can think about this. First is medical. So for people who do not have a proper connection between their brain and their muscles, so people with either people who were born with disabilities or people who went through traumatic brain injuries or spinal cord injuries or like just any or neuromuscular disorders that prevent their movement, we can provide a interface between their brain signals and something like a prosthetic leg, so that they can actually control it and regain their mobility in a sense. Other applications of this would be, I guess, in a more commercial sense. I guess you can also have you can drive a car with your brain, and I think there are research being done there by, I think, MIT. And you can also just control a computer with your phone, which just like thinking what you want to do with the computer. And I think a dominant example of this is what Neuralink is doing. I think they're developing a an integrated brain chip that controls a computer device.
Oh,
Yep.
wow.
I think your second question was whether we were planning on doing this further.
Yeah, further,
Yeah.
do you continue this?
Yeah. So for the four of us, I think we're all planning to continue with this specific research for like the oncoming semesters. And I guess I shouldn't speak for what they're planning on to do for like further into the future, but for myself, I think this is a topic that's really interesting. So this is, I guess, the career that I would want to pursue, like developing brain-computer interfaces.
Wonderful.
There is one more application that hopefully comes to market that you just tell to your 7 years old through your brain, that's time to stop playing game. That's time to have dinner. That's time to take shower. That's time to go to bed. So hopefully that interface will be there too. So yes, I think you have some more questions. You can come out of the mute, Ashwin, and ask your question.
Cool. So my question is, so you were talking about, okay, so from what I understood, you have to calibrate each person's like brain signals directly to like, because people are different, so different brain signals will mean different things. So does that mean like for every person who would want to use this, they would have to calibrate the machine to their brain like individually, or is there a way to do it where you like generally in the human brain, this signal means this?
Yeah, so I can explain this if nobody. Okay, yeah, I can just explain this real quick. So yeah, as you said, like everyone's brain is different in some sense. So since you do like end up having to like connect an EEG machine or some sort of machine to your brain anyways, it's not going to be an additional work to just calibrate it. Basically, you're going to be like thinking of stuff. So like I guess when you buy a new phone and you're trying to like get Siri to work, like you're gonna say stuff to Siri so that it knows how your voice works. It's pretty much the same way where you're gonna do some stuff so that the algorithm can know how your brain works. But like there's similarities, but some specifics to your brain so that the brain computer interface can do it perform its function with greater accuracy.
So Ashwin, just to explain that we don't do subject level analysis at all in the field of neuroimaging. We do group analysis, and that's why I bring all the brain images and signal, we bring all the brain images to a unified template. We normalize all the brain to the template, and then we look at the activity of the brain across the trial, across the time that, those activities that show the higher correlation across all the participants. That's a time that we can say that, when you are reading a book, this frontal region of your brain is getting activated, your IFJ gets activated. So yes, we never do subject level, and we don't, we do calibrate the machine based on the signal acquisition, but we don't calibrate the brain's activity that we get from participant prior, but we'll do it later as a group analysis, as when you're doing group analysis.
Okay, that makes sense. Thank you.
And I just want to answer the question that was in the chat. This data set was already published on Open Neuro. It's an EEG inner speech data set that was also described in a Nature.com article that was provided to us by Sahar. But they described the study and what occurred during the study, the participants, and then we conducted analysis on the data that was already provided.
Mm-hmm.
We can generate data if we have the recording machine, but our organization is not that big yet. Hopefully one day, right? hopefully one day we'll have neuroimaging machines to do the acquisition of data in-house. So that will be great. Maybe it's a time we collect donations for that. Like we'll be accepting donations for neuroimaging machines. So if you know anyone who wants to support future neuroscientists, yes, why not? We are available. Okay.
Yeah.
So yeah.
Yeah, like EEG is just to put it out there, EEG is actually one of the most cheap neuroimaging techniques, and that's probably like one of the reasons that it's one of the most popular neuroimaging techniques. But it's still really high in price, I think. So our study had 128 channels. Just as a reference, something like an 8-channel EEG would cost around $500. So, and it goes way higher with the number of electrodes.
Yeah, and then the machine, the software, each of the caps cost around like at least 500 to sometimes even like 3,000-4,000 if it is you know, it's a different, like the Wi-Fi related, the wireless caps. Yeah, so, but we can always start from scratch. So any donation welcome. But yeah, so hopefully one day we'll have in-house acquisition of brain signals. I think that's a wrap for us. Thank you so much, team. Thank you so much for all of you who showed up tonight to hear about the amazing research going on at ASDRP, in particular in my group and in my lab. So we'll hope to see you every Tuesday at the same time, same link. And I hope my team delivered some new things for you tonight to carry on and think about.
Wonderful.
Okay, have a good night, everyone.
Thank you.